Okay, so what we're going to do today is a combination. What this section is kind of a combination of organic chemistry and biology, again, like medicinal chemistry always is, but it intersects in a different way when we start talking about DNA interacting agents. And so we're going to talk about this, and you're going to see how important it is to understand the organic mechanism of how biology works, and obviously how important it is to understand biology in order to create new drugs. And so, as a side note, please do check Blackboard for announcements. I made an announcement about redoing problems, and do check on that. Okay, so we're going to move forward, and we're going to talk about DNA interacting agents. Okay, so DNA, we're going to think of it as another type of receptor in the medicinal chemistry sense. It's another type of thing for drugs to bind to, basically. So DNA is DNA, and what we're going to talk about is anti-cancer drugs. A lot of chemotherapeutic drugs work by interacting with DNA, and it's highly toxic. And so that's really good because you want stuff that's going to kill cancer cells, and you need to kill all the cancer cells if you can. You really don't want any cancer cells hanging around. And so the way that is really good to kill cells is to mess up their DNA. Now, it does two things. Number one, if you mess up their DNA, it's quite possible if you mess it up bad enough that they can't reproduce, so they can't make any more cells. So that's kind of the obvious thing. Number two, if you mess up a cell's DNA, a lot of times it will undergo what's known as apoptosis, where it kills itself. And so you can get the cell to kill itself by doing that. So that's great too. So basically, you really want this. Now, that being said, DNA is DNA. So bacteria's DNA and our DNA, the sequence of bases are different. Where some of the methyl groups on some of the bases may be a little bit different in terms of if you look at patterns, but chemically it's the same molecule. And so this is a little bit of a problem because every cell in your body, cancer cells or not, they have DNA that molecularly, or if we think about it, as is going to be made up of the same organic molecules. And really it's basically exactly the same if you think about it, considering that not that many genes are mutated to make a cancer cell, right? But there's a ton of genes, which means a ton of bases in DNA. And so because of that, they're basically exactly the same. And so when you kill cancer cells, you kill other cells as well. And so that's why you only use these anti-cancer drugs for something like cancer, where people are going to die if you don't treat it. So cancer cells, what they are, and so we could certainly do a whole class on that, is they are cells that are rapid and abnormally dividing. So in cells, basically, they tend to be less differentiated. And what we mean there is that when a cell in your body becomes something like a skin cell or a bone cell, it's very differentiated, right? A cell in your skin and a cell in your bone, you can imagine that they're very different, but they are they're very differentiated cells, and they are very unlikely to divide a ton. So your skin cells aren't really dividing a ton very rapidly. They aren't rapidly dividing. Similarly, your bone cells aren't rapidly dividing. They're just hanging out being bones, right? So if a cell rapidly divides, like cancer cells, a lot of times they're more what we like to call undifferentiated, or more like what we call progenitor cells in the stem cell community. And what that means is that they're cells that can um, be anything. They're more they're not committed to be a bone cell or a skin cell or something like that. They have this what you might call pluripotent, where they could be a number of different cell types. And so they're kind of how cells are in an early embryo, for example, or like early on in life. And so this is what cancer cells are like. So what this means is, so you're like, why are you going into this? Number one, it's important because cancer cells, because they can divide so much, they can grow big. They basically can cause people to waste away because they use so much nutrients. Second, because they don't think of themselves, if you will, as they don't present to other cells as, hey, I'm a skin cell, hey, I'm a bone cell. When they move around the body, when they do what's called metastasizing, if something is originally, say, a breast cancer cell and now started to mutate, or was originally an endothelium cell and now is mutated to be a breast cancer cell, and then it metastasizes, say, to bone, when that cell ends up, say, in somewhere like the femur or something like that, it can say to those bone cells, it could say, oh yeah, I'm kind of like you, and the bone cells will be like, oh yeah, we could see how you can be a bone cell one day. We'll accept you into our group. That's basically what's happening in a way less personified way. And so that's one of the things that's really important about cancer, because the last great mystery and the thing that we really need to solve in the future in cancer is metastasis. Right? We need to figure out how to stop metastasis, and that's where the work needs to go in the future. I'm going to be telling you about what has been done and what we're good at, but that's what we need people to do in the future. Okay, so because cancer cells are growing so quickly, this is something we try to take advantage of. They need a ton of DNA precursors because they're making a ton of cells, which means they're making a ton of DNA. So by they, now I am talking about a tumor. So that's a group of cancer cells. So this is kind of the thing, is that we want to, since they're feeding and they're uptaking stuff so much, we're hoping that the tumor, which is a cluster of cancer cells, is going to uptake drug molecules faster than something like my skin or my bone, which isn't metabolizing as quickly. Also, because they're reproducing, they're dividing so quickly, they are lacking some repair mechanisms. Sometimes they're kind of sketchy, which can lead to more mutations, which can lead to more carcinogenic changes, or the whole reason that they're dividing quickly is because there's a messed up repair mechanism. That's another cause for cancer. But either way, they're kind of like making a lot of times DNA repair mechanisms are slow. The enzymes that proofread and repair DNA are slow. And so we try to take advantage of that. So the cancers basically figured out how to get around that and keep dividing, but we try to take advantage of the fact that it's not proofreading by inserting some of our, what we'll see is that we're going to take advantage of that by inserting some of our drugs that get by the initial proofreading that isn't happening very well, and then end up destroying the cell. So when your body makes new cells, when it makes that new DNA, it proofreads it. And if the proofreading enzymes say this cell's got some messed up DNA, then the cell will kill itself, which is great, so that you can hopefully limit the amount of mutant cancer cells that you have in your body. That's great. In cancer cells, they're like, well, let's override that proofreading. We just need these copies now. And so we take advantage of that. Not all cancers do that, but a lot of cancers do. Okay. Oh, that's another thing is there isn't one mutation that's universal to all cancer cells. There isn't a universal set of genes. There's genes that are commonly implicated in cancers. There's genes that we'll look for to be mutants, and these are called oncogenes because they, when mutated, they lead to cancers. Onco is from the Greek word for cancer. And so we'll look for it, but there isn't one set. So what I'm trying to tell you is this problem is incredibly complex, and we know a ton about it, but it's just a really deep, um, complex problem, cancer and the causes of cancer and stopping cancer, and it's that's why the war on cancer started in the 70s, right? And we're still fighting it. So that's why it's so hard for science to deal with cancer. Okay, so basically what we're playing with here when we're making anti-cancer drugs is we're playing with drugs that we want to administer at a dose that's toxic to the tumors but leaves normal cells alive, right? And so I'm sure probably everyone in this class has known somebody or is related to somebody that's had cancer, right? And we know, I'm sure quite a lot of people know how much suffering that this causes, right? That chemotherapy, for example, can cause in people. And what's going on there is even then, what we're trying to do is we're trying to give people these toxic drugs at doses that are just killing the tumor cells. And so we already know, probably quite possibly from real life experience, how hard it is, what that line looks like, and how rough that line is. So the idea is that we're killing a fraction of cells, but we're going to kill a fraction of healthy cells too, which is why you lose things like your hair, right? Which are rapidly dividing, the cells that produce your hair are rapidly dividing cells. So they're also going to get preferentially killed by chemotherapeutic drugs. So that's why people lose their hair. And we'll talk more about this as we go on. Another thing is that single drug treatments are not effective. So effectiveness depends on dose and frequency of drug, like we always talk about, and single drug treatments are not effective. You'll always see this, that there'll be a combination therapy. Chemotherapy is a combination therapy, and the reason for this is that the tumor will evolve to resist. So this was introduced, combination chemotherapy, this was introduced in the 50s. And before then, one of the main chemotherapies, we already had radiation, which is also really can be rough too. But we already had radiation to treat tumors. But when we had a combination chemotherapy, it was very helpful. And so the thing is that tumors, like when we were talking before, we briefly mentioned antibacterials doing antibiotics and bacteria doing this, is that they're able to evolve a resistance. Essentially, they acquire a resistance. They start learning about the drug that you're giving. They figure out that they need to pump that drug out, and they start upregulating pumps that get rid of those that drug as soon as it goes into the cell. So the tumor starts figuring out how to detoxify itself and starts expressing proteins that help it detoxify itself. If you have, say, 3 drugs, then the tumor is a lot more confused and can't figure out how to fight it as quickly. So the idea is that you want 3 drugs that work in 3 different ways. So you're attacking the tumor on 3 different fronts, the cells in the tumor. And so that's one of the ways that you like to hit. one of the most common chemotherapeutics is going to covalently, and as an example of this, it's given here, is that one of the most common chemotherapeutics that we're going to talk about in a sec covalently interacts with DNA bases. And the tumor can figure this out. So it can covalently interact with DNA bases to really F up the DNA, basically, for, lack of a, well, there are way better terms, but I think for a very catch-all term. So it really messes up the DNA. And the tumor cell can figure that out. It's like, hey, you know what? This DNA is getting really messed up a lot. I'm pretty sure that we need to deal with this. And so they're like, okay, let's start upregulating our proofreading. Let's get back in here and proofread. And so what you need to do is inhibit the proofreading enzymes as well. So you need to mess up that copy of DNA and then inhibit the proofreaders so they can't proofread. So that cell gets made, and then it's like, oh, I need to die because I am too mutated. Okay, so now with drug interactions, there's a number of very common combination therapies for different drugs, for different cancers. There's very standard ones that doctors use. And when they're designing new ones, or they're adding to it, or they're subtracting, because you could imagine if you're on the same combination therapy for, say, 5 years, at a certain point, your cancer might figure something out. It might figure it out. And so then you need to start playing around again. Or you need to start playing around, something changes, you need to start playing around with the doses. Those are all things that happen too in patients. And so one of the things that's really important, because you're giving a bunch of extremely toxic drugs in a combination, you need to be really careful about drug interactions. So an example of this is, like, you don't want to give a drug that's especially toxic to the kidneys with another drug that needs the kidneys to excrete this. And we'll talk about this more, drug metabolism, probably starting next week. So we'll talk about drug resistance when we talk about drug metabolism. Tumor cells have multiple resistance mechanisms. They're really impressive. So they mutate the target. They can mutate the target. They can exclude the drug from entering the cell, or they can pump it out. It's not mentioned, or that's what exclude from staying in the cell means, is pump it out. Or they can destroy the drug. They figure out how to destroy the drug. And okay, so now we're going to go into basically what these chemotherapeutic drug targets are. And that's DNA. So DNA, I'm sure a lot of you probably know this, that in double-stranded DNA, the ratio of A to T and G to C is always 1. So you have these two strands. You have 5' to 3', and you have another strand going this way that's 5' to 3', and they each have bases, DNA bases. They have a nucleoside, they have a sugar, they have a phosphate group, and then they have this nitrogenous base here. And every time that you have an adenine across from it on the other DNA strand, you're going to have a thymine. Every time you're going to have a cytosine, you're going to have a guanine. These make 3 H bonds to each other, GC. Adenine and thymine make 2. And you can imagine, basically, from an organic chemistry standpoint, DNA is really DNA. And the way that in between the DNA strands, all life is now boiling down to some hydrogen bonding, is that in between this, you just have these H bonds, and this is what's giving your DNA its structure. So overall, it's going to be forming a double helix, right? And so what we mean is by a double helix is that the first strand here, this is the first strand, it's going to form a helix, and it's complexed because of its bases. These are the bases in between here. It's complexed with another strand, so that other strand runs in this direction. And it's in a helix too, so it's two helices wrapped around each other, and so that's a double helix. And so there's different spacings to the helix, and this is because the way that it's set up sterically and in terms of degrees of freedom, those two glycosidic bonds, and so that's what they're talking about. The base is here, and this is its sugar. So this is a glycosidic, this bond here, right there is a glycosidic bond. Here's a glycosidic bond. Here's a glycosidic bond. Here's a glycosidic bond. They're not across from each other. See how this bond and this bond are not across to each other. And the sugar, here's the backbone here, and the backbone is here basically. Because of this, there's going to be two grooves, the major groove and the minor groove. So you can see here how there's grooves. If you were to think of this as a block of molecules, there's space grooves. There's a space groove here and a space groove here. And so you can see it here, right? There's that groove here, and there's a groove here. And so this is where, when repair enzymes are proofreading, so when repair enzymes are proofreading DNA, the DNA is made, what they'll do is they'll come in here, they'll flip this base out from the DNA. So they take this base here and they'll flip it out and check it. They'll see if it H bonds with some part of its own enzyme, and they'll be like, yep, that's a T. Those are some of the enzymes. Other enzymes are going to look at the shape of the backbone, etc. But this is one of the most important proofreading enzymes, is going to actually go through and flip each of these bases out and check them. The bases tautomerize, and so this is something we can take advantage of. Basically, if we make a base analog, we can have it tautomerize in such a way that it H bonds in the same way with a DNA base. But what we need to know is that we need to have the spacing of the either 2 or 3 H bonds so they look like a DNA base. So what we can do is we can start to add isosteric mimics. So this is isosteres. We've learned that word before. That look like DNA bases. And they can mess up DNA structure. So this is something we can do to mess up DNA structure. So I would say all DNA is based on shape, but if we talk now about how DNA shapes in relationship to chromosomes, there is a ton of DNA in the cell. And this just blows my mind. I love this. In each cell, there's 1.84 meters of DNA packed in the nucleus. And there's like a bunch of other junk in the cell. Isn't that crazy? You can pack the DNA into the nucleus. 1.84 meters of DNA in each of your cell nuclei. It's crazy. Okay. You definitely don't want that to unpack all at once. That would suck. Okay. So anyways, the way that this works is you have this DNA, and you're going to roll this around chromatin. So these are these proteins that are going to help you pack the DNA. You're going to pack these fibers in a nucleosome, and this is all going to be packed into chromosomes here. And the chromosome will undergo various stages of unpacking. But what will happen here, as you can imagine, there is a ton of packing of this DNA. And so if you want to get a gene expressed, you need to go in here and enable the relationship between this histone protein here. So This protein is called a histone. The chromatin is the packed DNA that's going with the DNA packed on proteins that's going to go into chromosomes. So sorry, I think I misspoke a second ago. I can't remember what I said. So this is a histone here. And these histone proteins that the DNA is associated with, if you want to get a gene expressed, you basically need to come in here and have some helper proteins help unpack some of this to get that gene back out there, make that gene accessible. Bacterial DNA can be supercoiled, and this is really helpful because basically it helps pack, in this case, bacterial DNA can be in a circular genome. So bacterial DNA will be in a circle like this, and so it will pack into these different coils, and they'll coil on itself. It's really best if you've ever been around a telephone cord. This is the kind of thing, have you ever seen what a telephone cord does where it wraps all on itself? That's what's happening in DNA coiling, and that's what's happening here in the supercoiling. So what you can do is that if you want to change how these coils are, you cut the strands. Let's say that you had something that looks like one of these like stretchy bracelets. If you cut both strands, you can twist the strands in opposite directions and realigned, and basically you're going to end up, if you do that repeatedly, you're going to end up coiling the DNA up. And so this happens also in eukaryotic, like human cells. And so what happens is that your body, enzymes in your body called topoisomerases, it's really better from this slide, are going to be doing this cutting, twisting, in order to help the DNA get packed up so that it's not like a stretched out phone cord. It's like a phone cord where it's got all those basically it's not quite knotted, or it can be knotted, but it's all kind of wrapped up on itself. And you can imagine it's going to get even more wrapped up because it's like you've taken the cord and unplugged it from the phone and put it back together and this kind of thing. And so those enzymes are going to be twisting and moving DNA and wrapping it up. And those enzymes are super cool, and they are a great target for chemotherapy. So this is kind of an esoteric thing, is that they can also resolve some other beautiful topological problems that's esoteric and beautiful. Look at how the DNAs, can you see how this is forming a knot? This is amazing, right? The strand, look at the one strand. It goes here, and then the other strand goes on top, right? And then it wraps back around on top and under, and this strand under and over. And so those are probably originally circles that got broken as they were prepared for this imaging. This is electron microscopy imaging. So here, these are this is a circle, right? And this circle goes around like this on top. So it goes on top, under, around, over, then under here, around. Amazing. So that's enzymes that are doing this. And what this circle is, is that you can kind of see there that this is chromatin. So you can even see that it kind of looks like the shape earlier when we were talking about here, right? Remember how it looks like little beads, kind of? You can kind of see there that it's little beads. And so what this is actually, this is bacterial DNA, but the same thing goes for our DNA. What they do is that they look at this with electron microscopy. The way that you can do this is that you can associate it with uranium salts that are very heavy, and they deflect an electron beam. So the uranium salts associated with the protein in this compound, and then the histone proteins, the electron beam comes in, bounces off the uranium salts, and then you basically generate an image. So it looks kind of, and the image is generated in a similar way that you would generate any other kind of photograph, basically, at the end of the day. well, I would say nowadays, it's the way that you would generate an X-ray. Okay, so these topoisomerase enzymes are incredible. Hopefully, I've convinced you of that. And there is a number of topoisomerases. You need them to live because you need them to pack 1.84 meters of DNA into the nucleus of your cell. And so there's topoisomerase 1 and 2. You might have heard this called a gyrase, depending on who you took molecular biology from, if you took it, in the event that you took it. These are called in humans, in our cells, topo 1 and 2. And they do different things with DNA. And so we're going to talk about that in a breakout slide.